Greetings, y'all. So we're going to talk about photosynthesis today, and just like in cellular respiration and fermentation, right? Redox is an important part in photosynthesis, and redox is a process where electrons are released by an electron donor and then transferred to an electron acceptor. In oxygenic photosynthesis, we see the electron donor being water, H2O. This is our outside external electron donor. Our electron acceptor is CO2 from the atmosphere. I said our, didn't I? We're not doing this, but you know what I mean. We also have reducing power in photosynthesis, and it will be NADPH. We saw NADH in respiration and fermentation. NADH plays a big role in catabolic reactions. And in the last video, we defined this as being a process of breaking down large molecules. NADPH has a big role in anabolic reactions. And we define this as a process of synthesizing, making large molecules. And in photosynthesis, in a part of photosynthesis anyway, we have our CO2, we're converting that into glucose. So we have CO2 as, this is inorganic carbon, and it becomes reduced to a 6-carbon sugar glucose eventually. So we are doing anabolic reactions in photosynthesis. We, not us, I meant plants, my bad. But yes, so NADPH plays a role in that. We'll also see NADPH in other parts of photosynthesis too. Another thing to know is ATP. We saw this in respiration and fermentation as well. ATP is adenosine triphosphate, and this is what it looks like. This entire compound is ATP, and it has phosphate groups. Adenosine triphosphate has 3 phosphate groups, tri, meaning 3, right? And it also has adenine, which this is our nitrogenous base, and this is ribose, a sugar with 2 OH groups, 2 hydroxyl groups. Phospho and hydride bonds, these are high energy, quote unquote, high energy bonds that connect phosphates together. This is your phospho and hydride bond. This is your phospho and hydride bond as well. This is not a phospho and hydride bond. I made a mistake in the last video. That is my bad. Hydrolysis of ATP, that is the breakdown of ATP using water. We use water to break one of these phospho and hydride bonds, which releases a phosphate group. We get ADP, adenosine diphosphate, and an inorganic phosphate as a product from hydrolysis. Hydrolysis is also exergonic, meaning our Gibbs free energy, our delta G, is going to be negative. And this is coupled in other biochemical reactions within metabolic pathways. And we saw this with respiration and fermentation. We're also going to see this in photosynthesis as well. So reactions being coupled with each other is really important in metabolism, and it is a signature part of, I guess, like understanding all of these processes. Another thing to talk about would be the location of photosynthesis, which in plants, this is in the chloroplast. There are photoautotrophic organisms that can do photosynthesis without chloroplast, your photoautotrophic microbes, if you will. They can do photosynthesis without a chloroplast present, but for your discussions and for your concept maps, we mainly focus on plant photosynthesis, so I thought I'd add a little chloroplast here and go over the locations with you all. Y'all gotta do this for your concept maps anyway, so I hope this is helpful. We'll start with outer and then going in. This is your chloroplast outer membrane. This is the inner membrane of the chloroplast. Membrane. This is your stroma. This is the space here. Stroma. This is the thylakoid. They look like little coins inside the chloroplast. The membrane surrounding the thylakoid is the thylakoid membrane. And then inside the thylakoid is your lumen. So these are the different parts of the chloroplast that can be helpful just to know their locations and for your concept maps as well. Okay, so photosynthesis is made up of light-dependent reactions and light-independent reactions. We're going to start with light-dependent reactions first. And this is the process of converting light energy to chemical energy. And by chemical energy, I mean ATP and our reducing power NADPH. So this involves photosystems 1 and 2. These are complex proteins within the thylakoid membrane. So we have a location already. If I go back to the chloroplast, the thylakoid membrane is the membrane surrounding these coin-like parts of this organelle. In light-dependent reactions, we have light involved, right? Light from the sun. So photons, they attach to a molecule of chlorophyll. P680, we call this, in photosystem 2. This one is first. I know it's weird. Photosystem 2 is first when we have a photosystem 1 present. The naming in biology is interesting. But yeah, photons from the sun, they attach to a molecule of chlorophyll. You can find the chlorophyll within these photosystems, and they yeah. The electrons within photosystem 2 absorb these photons, making them excited. I should say, quote unquote, absorb. This is the process of photoexcitation. And if we were to talk about this in the context of the Z-scheme or in the context of energy level state, you can draw a diagram here. This is being your energy level state of the electron that we're talking about, low, high. The electron in photosystem 2 is originally at a low energy level state. When photons hit the photosystem and get absorbed by the electron goes from a low energy level state to a high energy level state, making it excited. So this excited electron within PS2, it makes PS2 very reduced, and so PS2 is going to give the electron away to other compounds, generating this electron transport chain. So this is in a series of redox reactions, right? So redox is important here, and we're seeing that now. The electron goes from molecule to molecule to molecule in a series of redox reactions. PS2, once it gives away its electron, it is electron deficient, and it needs an electron donor in order to resupply those lost electrons. And it does so in a process called water splitting. This is the process of oxidizing water into oxygen, which releases protons and electrons. The electron goes to PS2 so it can be replenished. Oxygen leaves as a byproduct of light-dependent reactions. Of course, in anoxygenic photosynthesis, right, we have a different outside electron donor. For some photoautotrophic organisms, they can use compounds like H2S, for example, as their outside electron donor instead of water. And that gives us a different byproduct that's not oxygen, right? Okay, so we have the first half of light-dependent reactions. Here's some more information about the electron transport chain. So the electron transport chain is powered through redox. There are mobile electron carrier proteins that help move the electron through the cytochrome complex and then to PS1. Your cytochrome complex, this is also a protein complex that uses the energy from the excited electron to pump protons out. And this creates a concentration gradient, right? It leads to proton motive force. We also have an ATP synthase in photosynthesis that uses proton motive force to make ATP, and this ATP goes to the stroma. So looking back, if we were to talk about this with our energy level state perspective, I'm going to rewrite it here. Energy level state, this being low, this being high. Oops. I want green here. So in PS2, we started with PS2 first. I'm going PS1. Our electron starts at a low energy level state. When it is hit by light, photons are absorbed and the electron goes from low to high in energy level. So this is the photoexcitation part that we were talking about earlier. This electron participates in the electron transport chain via redox reactions. As it moves through the ETC, the electron decays, quote unquote decays. What I mean by that is it decreases in energy level state over time. I forgot to put the cytochrome complex in here. Let's just say this is the cytochrome complex. It goes through the ETC, right? And it decreases its energy level state as it goes through this first leg of the ETC. When it reaches the cytochrome complex uses a lot of the energy from the excited electron to pump protons out, which this develops that proton gradient, which ATP synthase uses to make ATP. It then reaches photosystem 1, the electron, I should say, and it's back in its low energy level state again. So in order for this electron to continue moving, it needs to be re-excited. So we have to have some light hit it once again. Oops. That's in this part here. But we'll get to the diagram of the ETC in just a minute. And we talked about this earlier, right? So by the time the electron reaches PS1, it's no longer excited, and photons from the sun interact with the chlorophyll in PS1. This is chlorophyll P700, and the electron absorbs these photons from the sun and becomes excited again. So we see photoexcitation happen twice. And this makes PS1 very reduced, and so it's able to give this electron away to other compounds, which result in the second leg of this ETC. Although it is a very short leg compared to the first. And we have mobile carrier proteins, the most important one being Ferredoxin. which take the electron to an enzyme called NADP plus reductase. This is our enzyme that makes our reducing power, NADPH. And this also goes to the stroma. So let's look at the actual diagram now, which I have drawn for you. So we have PS2 first. Sunlight hits PS2 and it goes from pigment to pigment to pigment to pigment until it reaches this electron. The electron absorbs those photons, which make the electron become excited. In this process of photoexcitation, the electron goes from a low energy level state to a high energy level state. So let's just say that it's at a high energy level state. As it travels through this first leg of the ETC, it decreases its energy level. When it reaches cytochrome C, the cytochrome complex uses the energy from the excited electron to pump protons. This generates a gradient alongside the protons from water splitting, and we needed water splitting, right, to replenish the electron that PS2 lost. But these protons develop a gradient. ATP synthase uses that gradient to make ATP, which goes to the stroma. Of course, this electron eventually reaches PS2 and is back at its low energy level state. The sun hits PS2, photons go pigment to pigment to pigment to pigment until it reaches that electron in P700, and the electron undergoes photoexcitation once more. So it's at a high energy level state again, and it can move through the second leg of the ETC. The electron eventually reaches Ferredoxin. Ferredoxin is really important for photosynthesis. And this electron carrier takes it to NADP plus reductase, which makes NADPH, our reducing power. This goes to the stroma as well. So this is your light-dependent reactions. This is the entirety of it. Whew. And then we mentioned things from this slide. There is also another part to light-dependent reactions that is really important to understand too, and it is non-cyclic versus cyclic photosynthesis. In non-cyclic photosynthesis, we're using light energy to produce a little bit of ATP and reducing power in the form of NADPH. In non-cyclic photosynthesis, electrons do not cycle back to the photosystem. That means we need an outside source of electrons to come recharge the photosystem, that being water in oxygenic photosynthesis. So if we look at the ETC drawn out, this is your example of non-cyclic photosynthesis, in which you see the electron is not cycling back to the photosystem. It goes straight to NADP plus reductase to get that reducing power. That means our outside source of electrons, that being H2O, is needed in order to replenish electrons in PS2. So this is your non-cyclic photosynthesis. And I can rewrite the Z-scheme again. The Z-scheme is just referring to the energy level state of the electron as it goes through photosynthesis. And we saw this earlier, this energy level, this being low, this being high. I'm going to redraw the photosystems a little differently. I'm going to use the colors like right now. PS2, PS1, using all the colors I need now so I don't have to come get them later. This is our light. So the electron from PS2, low energy level state, right? When light hits it, photoexcitation occurs and the electron becomes excited. And it goes through the ETC until it reaches the cytochrome complex, which pumps out those protons, developing a gradient that ATP synthase uses to make ATP. But as it goes through the ETC, right, the electron is decreasing in its energy level state. And when it reaches PS1, it's back low again. That light hits PS1, makes the electron excited in the process of photoexcitation. It goes through the second leg of the ETC, where it meets Ferredoxin, which takes it to NADP plus reductase to get our reducing power. Of course, in non-cyclic photosynthesis, we need an outside source of electrons to come and replenish the electrons lost in photosystem 2. So H2O, O2. Right? And the electron goes back here. In cyclic photosynthesis, we're using light energy to produce proton motive force and then ATP from proton motive force. In cyclic photosynthesis, those electrons return to the photocenter, making it cyclic. So if we go back to our Z-scheme. Oh, and the reason why it's called a Z-scheme, or why we refer it as a Z-scheme, because if you see the shape of this, it does look like a Z in a way. So just another visual to help you. But yeah, if we were to redraw this, actually, let's use this. Just kidding, it just deleted itself. Okay, I guess we're gonna redraw it. If we were to redraw this, why did that just, oh my gosh, please. What happened? Okay. If we were to redraw this, this being our PS2, this being our PS1, we also need light. We learned that the electron goes from low to high when it absorbs photons from light at PS2, travels through the ETC, losing or decreasing in its energy level. So it's back to being low again. When light hits the electron, it absorbs those photons, makes it excited once more, goes through the second leg of the ETC to make NADP plus, not NADP plus, but NADPH. This was what we learned about in non-cyclic photosynthesis, right? In cyclic photosynthesis, we mostly make ATP. And so when the light hits PS1, that makes our electron go from a low energy level state to a high energy level state, we have Ferredoxin, our electron carrier, right? That takes this electron, brings it back to the cytochrome complex, where it pumps protons, right? Forming that gradient that eventually ATP synthase uses to make ATP. And then the electron goes back to PS1. So actually, In cyclic photosynthesis, PS2 is not involved. Oops. PS2 is not involved, but PS1 is. So this cycle repeats, and this is mainly used for making ATP. The electron, right, after photoexcitation is taken by ferredoxin to the cytochrome complex to pump protons, forming that gradient, proton motive force, that ATP synthase uses to make ATP. The electron goes back to the photosystem, and this repeats. And so because the electron returns to the photosystem, it does not need an outside source of electrons to replenish what it lost. Unlike PS2. PS2, as we learned, oh yeah, it deleted. PS2, as we learned, needed a source of outside electrons in order to be replenished. This is a non-cyclic photosynthesis. And that's because that electron went straight to this enzyme, NADP plus reductase, to make our reducing power. So in order for non-cyclic photosynthesis to repeat, we need that outside source of electrons to come resupply photosystem 2. But in photosystem 1, the electron itself is cycling, eventually returning back to PS1. Okay, so the most important part that I want to get out of cyclic and non-cyclic photosynthesis is the role ferredoxin has in both of them. I mentioned ferredoxin in both cyclic and non-cyclic. Ferredoxin is the one that determines if the electron cycles back or goes through non-cyclic photosynthesis instead. So ferredoxin is responsible for moving that electron to cyclic or non-cyclic photosynthesis. I also want to say in plants, right, cyclic and non-cyclic photosynthesis work together. In photoautotrophic microbes, cyclic and non-cyclic photosynthesis are separate. Just another thing to say there. Okay, so now we can get on to the second half of this video, which is light-independent reactions. And this is modeled by the Calvin cycle. The Calvin cycle includes carbon fixation, reduction, and regeneration reactions that eventually produce glucose. So we'll start with carbon fixation first. In carbon fixation, we have CO2 from the atmosphere that comes in. This enzyme called rubisco, I actually am not going to say the full name of rubisco, but this is an enzyme that helps fix our inorganic atmospheric carbon to organic carbon and has a role in producing sugars. So rubisco catalyzes the reaction between CO2 and this compound, ribulose bisphosphate. When these two bind together, it's highly unstable. And so they will break away to form two molecules of this compound, 3-phosphoglyceric acid. This is your organic compound. We went from CO2, inorganic, to 3-PGA, which is organic. This is what happens in carbon fixation. We're fixing atmospheric carbon to organic carbon to produce those sugars. Of course, we also have the use of ATP in order to do this. So we're using ATP that we made via ATP synthase in light-dependent reactions in order to make this reaction to get our organic compound. Now we have reduction, and it is reduction of the carbon compound. So reducing power is used, I should say oxidized. And 3-phosphoglyceric acid is turned to 6 molecules of glyceraldehyde 3-phosphate. One of these molecules is released out of the Calvin cycle and gets involved in another metabolic pathway that will eventually turn it into glucose. And glucose can participate in other parts of the plant as cellulose for structure or as starch for energy storage. So the plant cell is going to use this glucose in some way. 5 of these molecules, 5 of these glyceraldehyde 3-phosphates get regenerated back to this compound, ribulose bisphosphate. And we also need to use ATP here in order for that to happen. Boom. So as you can see, I did not specify each reaction in the Calvin cycle. You are not expected to memorize every single reaction that happens in the Calvin cycle. For the most part, you are just expected to know what happens in carbon fixation, what happens in reduction, and what happens in regeneration. Okay, just a side note about Rubisco. It has a high affinity for molecules like CO2, but it also has an affinity for molecules like oxygen as well. And so sometimes Rubisco binding to oxygen instead of CO2, because it doesn't really know the difference between the two, is a problem. When Rubisco binds to oxygen instead of CO2, which we find these compounds in our atmosphere, right? The Calvin cycle cannot work. We can't do the Calvin cycle if Rubisco does not bind to CO2, and so we can't get our sugar. For the most part though, just focus on Rubisco binding to carbon dioxide. You don't have to worry too much about Rubisco's affinity for oxygen, but it is an aside. It's just sometimes there's problems with Rubisco. But in any case, that is photosynthesis for you. We're gonna do a recap. We're gonna do a small little concept map of everything that we learned, starting with light-dependent reactions. So this is our light-dependent reactions. And let me put light-independent reactions here. Calvin cycle. Okay, just as we've learned using the Z-scheme and through that diagram of the ETC, light-dependent reactions involve photosystems 1 and 2, with photosystem 2 being first. When light hits this photosystem, the electron absorbs those photons and becomes excited. It goes from a low energy level state to a high energy level state. PS2 thus becomes very reduced, and it's going to want to give away this electron in a series of redox reactions that generate a electron transport chain. And so I'm just gonna model it by arrows. The electron eventually reaches the cytochrome complex, this blue one being the cytochrome complex, where it uses the energy from the excited electron to pump protons. And ATP synthase uses the proton gradient that forms from pumping these protons to make ATP. And this goes to the stroma. PS2 also needs to replenish its electrons that it lost. And so water splitting happens here. Of course, with anoxygenic photosynthesis, you'll use a different compound from water, something that does not produce oxygen as a byproduct. So the electron goes here. There are some protons in water splitting when we oxidize water into oxygen that participate in the proton gradient, which ATP synthase uses to make ATP. Some of that goes through here too, but I won't display that. The electron decreases in its energy level state as it passes through the ETC. When it goes to PS1, it's back in its low energy level state. Light hits PS1, which the electron absorbs, making it excited again. And it can now move through the second leg of the ETC where it meets Ferredoxin. Ferredoxin takes it to NADP plus reductase that generates our reducing power. And that also moves to the stroma. This is our non-cyclic photosynthesis. In cyclic photosynthesis, we're making ATP. There's no reducing power that's being made in cyclic. We have the electron, once light hits it in PS1, gets excited. Ferredoxin moves it to the cytochrome complex where it pumps protons, which ATP synthase uses to make ATP, and then it cycles back to PS1. This is cyclic photosynthesis. But what I have drawn here is non-cyclic. For plants, cyclic and non-cyclic photosynthesis work together. In the Calvin cycle, we are using the ATP and the NADPH generated from light-dependent reactions in here. So let's make the Calvin cycle. Ribulose bisphosphate is involved here, but our input is CO2. And in the Calvin cycle, right, we have 3 parts: carbon fixation, regeneration, and reduction. That is not in order, but we start with carbon fixation first. Carbon fixation. Rubisco helps catalyze the reaction between ribulose bisphosphate and CO2, which is highly unstable, and so it's going to break apart to form 2 molecules of 3-phosphoglyceric acid, our carbon compound. So we went from atmospheric inorganic carbon to organic compound to help produce these sugars. That's what happens in carbon fixation. We need ATP, so we use ATP here. In regeneration, in reduction, sorry, that's our next part of the Calvin cycle. 3-phosphoglyceric acid, that's going to be reduced to 6 molecules of glyceraldehyde 3-phosphate. So one of these is released and goes through another metabolic pathway in which we get glucose. 5 of them get regenerated. That's the next step. While we're reducing 3-PGA to G3P, we need oxidation because reduction is coupled with oxidation, and so we oxidize NADPH to NADP+. This glucose that gets released, it can participate in other parts of the cell as cellulose for structure or as energy storage and starch. 5 of them, again, get regenerated to form ribulose bisphosphate again, and the cycle repeats. In order to do this, we need ATP. So we need to use ATP. ATP to ADP. So this is your Calvin cycle. Notice that these two need each other. Let me put this in another color. We got ADP from the Calvin cycle, which ATP synthase takes. We got ATP from light-dependent reactions, which the Calvin cycle uses here. We got NADP+, which our enzyme NADP+ reductase needs to get our reducing power for light-dependent reactions. We also got NADPH from light-dependent reactions, which the Calvin cycle uses. So you can see that the products of these two parts of photosynthesis interchange, and they are a part of each other. They need each other in order to happen. So this is photosynthesis for you. If you have any questions, please feel free to reach out. I wish you nothing but luck in your studying and your exams. Thank you so much for watching. Bye-bye. Oh, I forgot about this slide. My bad. Lol. In oxygenic photosynthesis, your ultimate electron acceptor is water, and then, ultimate electron donor is water, and then acceptor is CO2. In anoxygenic photosynthesis, right, our outside electron donor is going to be different from water. But yeah, anyways, that is all I have to say. Thank you so much for watching. Good luck in everything that you do.